after a lucid lecture from Dr. Deepak Jomani sir, uh, we are here for another topic on lactate physiology. And before I start the talk, I really thank Dr. Ashutosh sir, Pallavi ma'am, Diabetic, for giving me this opportunity and this honor of presenting Dr. Anup Mishra oration. And this is his credibility, and I am honored that this particular talk has been given to me in his name. Thank you so much, Diabetic and Mishra sir and madam. So we will be talking about the lactate revolution, how it has changed from waste product to therapeutic target in diabetic atherosclerosis. Since medicine, we have tried to understand the medicine, we have been dismissing the lactate as a metabolic waste product of anaerobic glycolysis, and it has been viewed negatively, leading to lactic acidosis in situations of tissue hypoxia and a marker of poor prognosis. Recently, this is considered as a signaling molecule and a fuel source. We know that in diabetes, there is increased glycolysis which increases lactate due to insulin resistance and mitochondrial dysfunction, and the increase in lactate levels by 2 to 4 times, which is common in uncontrolled diabetes, is also suggestive of diastolic dysfunction in cardiac conditions in diabetes. The atherosclerosis connection of lactate is directly correlated to the plaque burden of the carotid arteries and is independent cardiovascular risk marker, which is reflection of myocardial dysfunction in the vessel walls. Increase in lactate production leads to increased reactive oxygen species, which leads to endothelial dysfunction and vascular smooth muscle proliferation. In anaerobic glycolysis, there is more of formation of foam cells, which is leading to the increase in the plaque size and ultimately drives the progression of atherosclerosis. Lactate is considered as extracellular signaling molecule via GPR81, that is G protein coupled receptor 81, also known as hydroxycarboxylic acid receptor. In physiological conditions, this is beneficial and is expressed in adipocytes, skeletal muscles, liver, kidney, immune cells and vascular endothelium. It couples with the GI protein, inhibiting the adenyl cyclase, which reduces the cyclic GMP. It activates the protein kinase C and A within 5 to 15 minutes, and downstreams lipolysis, anti-inflammatory response is there, and the vascular modulation is occurring in physiological condition. But when there is accumulation of lactate, there is inflammation, oxidative stress, calcification of plaque and instability, because the decreased lipolysis or decreased free fatty acids or the anti-inflammatory properties, the protective effect of GRP81 activation is lost, and this is probably because of the narrow therapeutic window of GPR81, which is in terms of its concentration, duration as well as tissue content. The increase in lactate therefore can lead to inflammation and angiogenesis, and it can also be anti-inflammatory in physiological condition. Here comes the concept of lactylation, which suppresses the inflammatory mediators. We know that glycolysis increases lactate, which down regulates the medium chain triglycerides, and the vascular smooth muscle cells phenotypic switch is activated, which causes mitochondrial dysfunction, therefore proliferation and calcification of the plaque. Lactate has therefore been understood as a signaling molecule because it physically binds to specific protein sensing G protein coupled receptors, translating extracellular metabolic fluctuations into precise biological response, is expressed highly in adipocytes, immune cells and endothelium, regulates the lipid metabolism, angiogenesis and vascular tone, and is expressed in macrophages, which is a role in plaque formation and through foam cells. It also acts in the macrophage system, where also it activates the inflammation and the plaque progression. We also know that free fatty acid is an independent atherosclerosis factor which is derived from the lipolysis of adipose tissue. Lactate in physiological conditions activates GPR81, which suppresses lipolysis, and therefore the free fatty acids are reduced by almost 40% without any side effects. Now, lactylation, what it is? It is new post-translational modification which has recently been understood through histone or non-histone levels, and it helps to regulate the inflammation, angiogenesis, and therefore calcification of the plaque. In aortic valve disease, there is increase in lactate which increases the histone lactylation and therefore drives expression of the calcific genes, therefore accelerates the progression of the disease of the valve, aortic valve, its calcification. It promotes the macrophage polarization in atherosclerotic plaques, therefore enhancing the calcification. In diabetic myocardial infarction, the lactate levels are almost 40 to 60% higher in comparison to non-diabetic individual, which also translates into increase in heart failures, arrhythmias and cardiogenic shock, and this is happening because of the reduced lactate oxidation and accumulation of the cytosolic lactate. So, lactate can lead to atrial fibrillation, heart failure, myocardial infarction, atherosclerosis, and this is happening to the smooth muscle cells and macrophages and monocytes. A 24-hour lactate concentration is directly related to the mortality in diabetic cardiovascular emergency, and improved clearance of lactate has shown up to 50% reduction in the mortality, and the serial lactate monitoring has been found superior to the one-time measurement of the lactate levels. Metformin tends to increase the lactate production through mild complex 1 inhibition, which tends to activate the GPR81 signaling, and we know that this activation, we have seen in previous slides, has cardioprotective benefits by mobilizing the lactate and utilizing them. Therefore, uses of metformin improves the CV outcomes as well as lowers the glucose levels. SGLT2 inhibitors acts by shifting the cardiac metabolism towards ketone and lactate utilization, and therefore it improves the myocardial lactate oxidation efficiency in diabetes, and therefore we have cardiovascular and renal benefits through SGLT2 inhibitors. Because of the narrow therapeutic window that at particular level there is GPR81 activation, there is no harmful effect. But when the levels are higher, more than 5 millimoles, there is accumulation of lactate which is harmful, and this targets the vascular and adipose levels, and therefore we need to differentiate the physiological and non-physical. That therapeutic window is important. Now, so at present researches are on, and we are looking forward to the uses of these agonists in diabetes and established atherosclerosis for synergistic CV benefits along with SGLT2 inhibitors. The inhibitors of lactylation are useful, should be useful in calcific atherosclerosis and vulnerable plaque stabilization. Therefore, its production and clearance should be balanced to have the benefit. The duality of lactate helps in the polarization of macrophage, which dictates the functional phenotype of macrophage and microenvironment. The lactate clock in acute inflammation through histone lactylation suppresses the pro-inflammatory genes, and therefore homeostasis is activated. The pathological duality of this molecule, where physiological inflammation, the chronic lactate accumulation can force macrophages into maladaptive, senescent, chronically active states depending on the context of the disease. It has been recently studied since last 5-6 years. Lactylation through lactyl coenzyme A group added to lysine, both to histone and non-histone groups, is helpful in understanding the gene regulation. The lactate is converted to pyruvate lactate dehydrogenase B, which enters the TCA cycle in mitochondria via lactate oxidation complex and serves as the precursor for gluconeogenesis and substrate for protein lactylation, and therefore regulates the gene expression. In atherosclerosis, lactylation tends to increase the glycolysis and LDHA activity, therefore the gene expression, and it reduces the vascular inflammation and therefore therapeutic target in atherosclerosis. It can be used as treatment in terms of 3 levels: production, transport and epigenetics. Niacin has been found to be useful as GPR81 agonist, which reduces adipose lipolysis and therefore reduces circulating free fatty acids by 40% and therefore reduces the progression of atherosclerosis. Other molecules in this category include ionidamide, dichlorate, and these are under glycoflavine, vitamin C, stiripentol, which are as of now in research stage. Second is enhancement of oxidation of lactate through oxidation, which improves myocardial lactate oxidation in diabetic hearts and therefore restores metabolic flexibility, increases lactate uptake and utilization, and here the molecule uses dichloroacetate. Other molecules are also there like ibuprofen, salicylic acid, mepalazumab. Then there is lactylation inhibitors, which reduce calcification gene expression and reduces inflammation in early preclinical stage, and here the molecules like curcumine, sodium butyrate, epicidine, etc. are being used along with nicotinamide. So to conclude, lactate has progressed from metabolic waste product to signaling molecule and therapeutic target. In diabetes, increased glycolysis increases lactate levels, which leads to endothelial dysfunction, vascular remodeling, macrophage polarization, which causes plaque progression through inflammation. Lactylation is a metabolism condition which regulates the gene expression in atherosclerosis. It can be used as a novel therapeutic target, this lactate axis, and is helpful in diabetic atherosclerosis and therefore the disease progression. GPR81 agonists are immediate transitional opportunity for free fatty acid reduction and plaque stabilization, and it can be complementing to the existing medical therapy in CKM syndrome. Thank you so much. Any questions? Because this is as of now a research molecule. Yes, ma'am. This can be understood as one of the salts of lactate, and that is again a marker of inflammation. We see whenever we are doing cardiac markers, LDH is one of the cardiac markers along with troponins and CRPs which we are seeing. So probably these are the sources from where we could understand that this could be one of the therapeutic targets in management in such situations. Thank you.